about Dr. Bolaji again. So Dr. Bolaji actually attended Queen's University in Kingston, Ontario, and she pursued her undergraduate degree in biology and graduated in 2014. She specialized in cell biology and genetics, and in July 2014, she actually got a scholarship to pursue a doctorate degree also in cell biology, cell and molecular biology. She finished her PhD in 2019 and she worked as a postdoctoral researcher at the University of Manitoba between 2019 to 2021. Right now, she's an associate professor at the Department of Biochemistry and Medical Genetics at the University of Manitoba. She's also working as a computational biologist at National Microbiology Laboratory in Winnipeg. Right now, too, she's also an adjunct faculty at the Providence University College. And she got an award in 2023, the Public Health Agency of Canada Award in Applied Sciences and Research. She is actually enthusiastic about research and about using computational tools to solve difficult problems. Also, she enjoys mentoring young students and scientists. She's the founder of Lead Within. Lead Within is actually an NGO that actually meets with youth at her local church in Winnipeg, and they organizes career workshop for people. Whenever she has some time, she enjoys going on long bike rides with her husband. So, Dr. Bolaji, you are welcome once again. You can now share your slide.
Again for the warm introduction. So we'll get into it. Odmayo has done a very nice job introducing me, but I like to think that when I talk about my story in bioinformatics or even my career so far, it's often best to start from the very beginning so that people can know where I'm coming from. I have a slightly different computational biology background on the academic side, and it would also give those who are hoping to venture into such field an idea of how to get in. Also let you know that more often than not, as it's said, your background doesn't necessarily determine your destination. So just to reiterate what has already been said, I will start, first of all, walk you through my educational and professional background and then throw in how bioinformatics or computational biology became a thing for me. As they've said, I graduated in 2014 with my undergrad degree, so just a little over 10 years ago now. And why I like to highlight this is that in 2014, for those who have been in the computational space or bioinformatics space for that long, you would realize that in 2014, there was very little about it. If people even spoke about bioinformatics like they did during my time, it was R. They were essentially telling people that, oh, you need to know how to code in R, or you need to know how to analyze your data in R, or use the statistical packages in R so that you can have a career at the end of the day. And I would say I caught that bug because in my second and in my third year of my undergraduate degree, I took an R course. It was good, but it wasn't necessarily useful for me at that point. And so as things progressed for me in the research field, I knew I wanted to do a PhD for a lot of reasons, but one of the most glaring reasons for me then was that I didn't want to be told what to do. That was something I knew at a very tender age, that I wanted to be the boss of myself. And so I got very close to a professor who ended up mentoring me. And she told me, when she said, you're kind of good with research. What are the things you see as your strong points? And I said, well, I know I like research, but I'm not really sure I want people to be telling me what to do because I feel it can constrain my creativity. And she said, well, then you should be a professor because as a professor, you tend to choose all the research you want to do. And if you don't kill anyone, you're always going to have a job. So then I sat down and I asked myself, what's the quickest way to get to that point, to be a professor without having to stay in school forever? As God will have it, my grades and everything aligned. There was an opportunity that opened up in the French-speaking part of Canada. For those who aren't too familiar with Canada, Quebec. So I moved to Quebec in 2014, just shortly after my bachelor's degree, and I started my doctoral studies there. It was a very eventful time in my life, but then again, I had to built from my strength. My strength was in the wet lab side at that point. It was in cell and molecular biology. Permit me to highlight that during my undergraduate days, I had the opportunity in volunteering and working in a total of 7 labs. So when you're talking about wet lab skills, I had very good hands, like I was often told. So good hands, my pipetting skills were top notch and things like that. So I built on that during my postdoctoral studies in the same university, in a different university, pardon me, and but in the same field, cell and molecular biology. It was heavy on plant biochemistry. So I put up a very typical figure of the kinds of research we were doing. It was very heavy on the wet lab side. We were essentially, my goal then was essentially to compare protein-protein interaction for defense in plants, focusing mainly on RNA silencing. So this helped me then to understand, virology, to understand viral genetics, and also to understand how biochemistry assays were done. So we did a lot of immunoblotting, a lot of Western blot, a lot of Northern blot, a little bit of PCR and all of that. And my major PhD project was actually to clone different genes from one plant to the other. I don't want to get into the nitty-gritty of things, but I was essentially trying to clone a gene, pardon me, that was encoding for a protein that conferred resistance in tobacco to a wide, host of viruses. I was cloning it from tobacco into tomato. It was a pretty interesting project at the initial stages. It was rather challenging too, because for those who are familiar with working with plants, you're working with diploid, sometimes even haploid, sometimes even genes that have 4 copies. So you have to account for so many things. I wouldn't say it was a walk in the park for me. It wasn't a walk in the park for me. And in fact, I included it here that at the end of the program, so I was in this program for about 5.5 years. At the end of the program, I was mentally exhausted. I like to call it fatigue. Not necessarily because the project was a bad project. It wasn't a bad project. In hindsight, it was a very good and a very thorough project, but it was difficult to get results consistently. Those who are in the wet lab space might be able to attest to it. One experiment took several days. So, you got to wait for the plants to grow, you got to extract your protein, you have to do this extract to RNA. There was a lot to do. And sometimes the results were very unclear. You would see a band today, you run the same experiment tomorrow, your band isn't there. Or sometimes you're like, oh, is this the right size? Or is this how big my band is supposed to be? There were so many things that discouraged me at the end of my PhD, I like to say my PhD journey, but it didn't end there. So I like to tell people that what really worked for me was honesty. I had done this, PhD. I had got into the stage where I was supposed to be an expert in the field, in the set field. Anyone who is doing a PhD or who has done a PhD would tell you that, you know your thesis inside out pretty well. However, at the end, I knew this was not what I wanted to do. Having spent 5.5 years, I wasn't finding satisfaction. I was mentally fatigued. Relationships on the professional side were tensed. So many things weren't going the way they should have gone. But I didn't look on that. I went online and started thinking about how can I use what I have to get to where I really want to be. And where I really wanted to be at that point was in the computational space. In fact, I remember talking to my PhD supervisor way back then that I wanted us to do some RNA-seq study. We're doing a lot of protein-protein interaction on the biochemistry side. So I said, let's see what's happening on the transcriptomic level. And he didn't have funding, but he also wasn't super interested in that kind of study. So he shut it down. So one of the things I told myself was, if I have a chance once I'm done, I'll try to get into that space. But then again, my background was wet lab, heavily on the wet lab side. I had mentioned earlier that I took a few R classes during my undergraduate studies. So some of you might want to know, how did I navigate that trajectory? Well, one of the things I did was I signed up for a class in one of the universities here, McGill University, if you're familiar with it. So it was a 2-semester course of computational biology for graduate students. So you come with your data, or they gave you data to work with, and that kind of threw me into the deep. The good thing was they were doing everything in R, so I already had that background to work on. So before I switch to how I made that transition into the computational biology space, I like to tell people that I don't necessarily see my PhD as a wasted time in my life, because there were a few, gratifying points about it. It was interesting. I learned a lot of new things. Most importantly, I learned what I didn't want to do. Unfortunately, it took me a PhD to figure out what I didn't want to do, but at least I did figure that out. Then on the wet lab side, I became quite good. And this ended up, as you would see later on in my talk, this ended up being an advantage for me. I got the chance to develop, lots of soft skills that are still very pertinent to the position I'm in now. As any PhD student will tell you, you have to write, you have to figure out how to write, and you present your findings in conferences, workshops, and whatnot. So during the course that I mentioned that I took at McGill University, it expanded my data analysis skills, and it also made me get into the phase of figuring out how I could set myself apart from other applicants. Because once you're done a PhD, you kind of have to face the reality of what's out there. And by the time you check out what is out there, this was 6 years ago, the competition was very stiff. I can only imagine how stiff the competition is now. So I wanted to explore something a bit more data analysis oriented, and I got into the postdoctoral program at the University of Manitoba. And how did that happen? Well, I'll just be very quick about it. During the end of my PhD, I had put in for a conference. So I put in for a conference and I presented my work, my wet lab work at that conference. One of the keynote speakers was just in the market for a postdoctoral researcher. He saw all the hard work I had done, and we started talking, and I told him, I want to do, some computational biology work. Him, on the other hand, at that point had money. He was also thinking about building some computational biology into his research work at that point. So he needed someone who was independent, and he told me he didn't necessarily need someone that knew it all. So he wanted someone who was independent. He had the money, and he needed someone who was willing and interested in that kind of a thing. So I would say everything aligned. I had the interest, I had the fortitude, and I like to say a PhD has given you or gives you the skills to be able to do anything in the research world that you set your heart into. So that was kind of how I transitioned into my postdoctoral role at the University of Manitoba from October 2019 till around April 2021. And so when I was there, you might want to ask, what exactly did I do? This professor gave me the autonomy, he gave me the independence to run a research project that I was very interested in. I was essentially interested then in running the whole genome sequencing platform of the university at that point. So we got our first Oxford Nanopore sequencer, and he also gave me the permission to get undergraduate students and graduate students who did the wet lab. So again, because of my wet lab experience, I could set up experiments in such a way that I knew how they would work, and I could help troubleshoot things. So I had that background where I trained the undergraduate students and the graduate students within 2-3 months. And so they were just chugging the data out for me, and I could focus on data analysis, but the learning of the data analysis, because if you followed my story so far, you would know that I haven't necessarily worked on my own data for computational exploration. It had been people's data in the course that I said I took, here and there. So I got exposed to that. I also got exposed to writing grant proposals, writing technical papers, and also writing publications, like articles for publication. And then, like I mentioned, I hired a bunch of other graduate students and graduate students. So that helped me on my administrative and HR skills. So what did I really do in terms of computational biology? Pardon me, I use the term computational biology and bioinformatics very loosely in my talk. So essentially, what I did for this during my time as a postdoctoral fellow was I focused on metagenomics projects. There was a metagenomics project that we were looking at. It was more on the plant level, plant microbe interaction. We wanted to identify microbes. So in our case, bacteria and fungi that would enhance the growth of plants without using chemical fertilizers. And we had funding from a company that wanted to eventually commercialize our product. So I led the project using long read nanopore sequencing technology. We identified novel microbes. I was a team member on numerous transcriptomics projects and functional genomics projects. And by the end of my 14 months, even before the 14 months, but within 14 months, I was able to publish a first author manuscript. And I will just give you a very quick overview. I know we only have, 15 minutes to go over this talk. So if you're interested, you can check this out in Frontier in Microbiology. It was a microbial community dynamics. We did a lot of beta diversity and alpha diversity, came up with PCAs, the phylogenetic diversity graphs, and relative abundance. I'm showing you this so that you can see that all of this is achievable. I'm coming from a heavy wet lab background. I had never created figures like this with my data. And then towards the end or nearing the end of my postdoctoral term, I was able to get figures like this. So the postdoctoral phase was relatively short, not because there wasn't funding, but because I had a really good mentor in my boss or in my supervisor, where middle of 2021, he told me that he felt I already had enough skills to venture into the computational biology space. And for those of you who are in the academia setting, you would know that postdoctoral researchers don't get paid much. So he said, it doesn't feel I'm getting paid for the amount of work I put in. I would like to highlight that all of that I achieved when I was a postdoctoral fellow, I still worked like I was a PhD student. For those who are really interested in work-life balance, I wouldn't advise you to do that. Going into the lab 7 days a week if need be, working up until the midnight, all of that. I didn't think because I had my PhD I could start coasting, because I knew this was a relatively new field and I had a lot to learn. I had to understand my data and know how to clean the data. Essentially, I had to figure out everything because my PI was also relying on me. This was also new to him, right? So I needed to be the subject matter expert and I had to do that very well. So thankfully, he saw the hard work I put in and he also noticed the growth that I had achieved in less than 2 years. So he encouraged me. I don't have a formal educational background in bioinformatics. So he said, you know what? There are a few jobs in the government that they're looking for computational biologists. Apply. I'm like, well, my PhD is in cell and molecular biology. I've only done, 1.5 years of formal or professional bioinformatics work. How do I convince these people to hire me? And he said, you know what? Just, leave that to me. I'll help you with your CV. So he helped me with my CV and my resume, helped me with my cover letter. Most of these jobs had written interview, oral interviews. So before the written interviews, he would prep me. And then before the oral interviews too, he did prep me, which I'm very thankful for. So by May 2021, I got a research scientist position with the government of Canada, but it was with an agency called the Canadian Food Inspection Agency, where they were just starting to get into genomics. So essentially what they wanted to do then was they wanted to look at how zoonotic organisms, were affecting the landscape. And for those of you who are familiar with timeline, this was around the heat of COVID too. So the old zoonotic genomics was on the rise. So I'll give you a quick overview of what my portfolio looked like there. So when I got to CFIA, that's what I'm going to call it for short, they wanted me to do half and half. So half wet lab and half computational work, which I didn't mind. Again, because it was in the heat of COVID, we were all taking turns in doing experiments in the lab. So it was perfect. On days where I had to be in the lab, I could quickly do my experiments. I could set up things on the sequencers. And then on days when I wasn't in the lab, I could access my data on the cloud and, do the analysis from there. So during my time there, you probably noted this was very short. It was a 5 month period. I conducted collaborative genomics projects with the aim of identifying novel zoonotic diseases using high throughput sequencing technology. I was also fortunate enough to work with a bunch of graduate and undergraduate students too. During my time then, I supervised their research work. I had the chance to still write papers and grant proposals, even though I was only there for 5 months. And there's always the administrative and HR portion of your work. Once a PI or the group leader sees that you're driven and you have that skill. So thankfully, I had someone who identified it then, and it was also able to give me all I needed to progress and to grow. Again, although I was there for 5 months, I would say this was the first time I was in a space where money wasn't the issue and they had all the technology. So we had all of these devices in addition to, okay, for those who are not familiar, let me walk you through it. So this is the nanopop Promethean device. It's one of the most powerful high throughput technology out there for long read sequencing. You can sequence so many things within a short period of time. The coverage, the depth of your genome or whatever it is you're sequencing is top notch. After which you have your gridiron, almost as good as Promethean because the flow cells can be used interchangeably. You need high performance computers to, more often than not, you need high performance computers to run these. These already have their inbuilt computer. So all you necessarily need sometimes is a monitor. They both run on Linux machines. And then you have these other MinION devices in different range, and you have a Flungal. Needless to say, they wanted to push this technology out because their idea was for researchers in the field to take their samples from the field and do the data analysis in the field and get the data back in real time. So we were building the technology for that. And in addition to the nanopore technology, they also had Illumina sequencers. So they had the iSeq and they had the MySeq, which was good because then we could do hybrid genome assemblies and also take advantage of the accuracy that Illumina data or Illumina technology gives over the long read technology. So my time there was highly productive. I also, got experience in analyzing data for comparative genomics purposes. I authored a manuscript there, a first authored manuscript there within my short period of time where I compared genome analysis of a certain virus, a coronavirus, from the very first Canadian reptile mortality event and compared it to similar viruses. So did analysis, add graphs and phylogenetic tree that could compare the different viruses that were obtained from different sources, and then looked at different portions in the genomes that were identified as recombinant hotspots. So this again expanded my horizon and made me realize that I really enjoyed the computational side of things. So by my 5 months in, I wasn't necessarily a fan of the wet lab side, and I wanted to explore an opportunity that gave me just 100% computational biology. And so that's how I got to where I'm currently at. So in October 2021, I got hired in the Public Health Agency of Canada, still with the government, but in another department as a computational biologist. Within that position, my portfolio expanded where they made me the genomics lead, working very closely with the Manitoba Provincial Lab. So in Canada, we have provinces just like states. For those of you who aren't familiar with that system, so I am the genomics lead of the provincial lab. And then, as Odu mentioned earlier, over time, I've been able to work my way back into the academic space, and I'll talk about it very quickly, just so that I could also have some independence in the kind of research I do. The government labs are great. You don't have to worry about funding for most of the things, but your research has to align with their mandate. So you don't really have too much flexibility for independent research. So currently, I've been in this role now for a little under, a little over, however I want to look at it. 4 years, going on 5 years, I believe. And essentially, what I do is I manage the provincial lab's computational biology units. Right now, I spend most of my time enhancing interprovincial, national, international data sharing. Everybody wants everyone's data. We can't just take people's data and throw it around. We have to go through the right mechanisms. We have to come up with material transfer agreement because data is, private. We're in the public health space. We're working with people's data, personal health information, things like that. Then I also spend a bit of my time improving bioinformatics workflow for optimizing disease surveillance. We do a lot of surveillance for respiratory pathogens. So it changes as the year goes on. Right now, respiratory pathogens are on the rise because of the way the season is structured. And then, in the winter, we then look at, things like enterics. So those who Well, that happens all the time, but we usually see spike in enterics. Then sometimes we also have to monitor HIV data, HIV genomics, and things like that, syphilis, all those things. So anything you can think of that could be of public health importance and that has some genomics importance, we do that. Then I help with troubleshooting on the wet lab. So that's where my wet lab experience still comes in play sometimes. My team and I look at the data. It's not looking like it should look like. Maybe the depth isn't good enough or coverage is not quite where it should be. I get on the phone with the wet lab leads. I walk them through with troubleshoot to get the kind of data we need. Because for most of you who are in the computational biology space or who have worked on some bioinformatics data, you would know that garbage in, garbage out. So this helps us to reduce overall sequencing and data analysis turnaround time. And then lastly, I build capacity for analyticals and rapid identification of COVID. COVID is still, a huge thing in the public health space, enterics, zoonotics, and viruses. So I'll just highlight a few of the things I've done so far. So far, upon everything I do, I've still been able to identify a space for publication. So last year, I published a work that I did all in silico. So in silico analysis identifies sequence contamination threshold for nanopore generated SARS-CoV-2 data. It was published in PLOS Computational Biology last year. One of the things I spent a lot of my time doing within the past 4 years is procuring high performance computers for the provincial lab. So writing the grant, writing the justification, and building a genomics network, in-house genomics network, because before they hired me, they didn't have any computational biology tool at all. They had a MySeq, but even the MySeq data, they sent it out for the data to be analyzed. So since I've arrived, we've been able to buy 7 high performance computers. In addition to the storage that we need in-house, I've been able to network everything, and we've worked with data architect on how we can allow seamless data transfer so that we can eventually get things to the end users. Because sometimes we get samples from clinicians, from doctors, they've taken a swab from a patient, they don't know what it is. We do the analysis to be able to identify what it is and send them a report that, okay, your patients could have this bacteria, or they could have this virus, or they're working with, or they have a bacteria, that cannot be cultured. And that's why the traditional microbiology tools hasn't been able to identify it. Then we've also been able to implement an in-house next generation sequencing information management system, is a mouthful, that I designed. We've been able to implement that so that we can have 2 separate systems, again, because of data privacy and also hackers. So they have a system where all the information of everyone in the province is on. We built another system for the genomic side. So once we have samples that we've worked on genomically, we don't want it being on the system that everybody's information is on, so that people don't hack it, because sometimes hackers, they've done that in the past for other provinces. So we're very conscious about data integrity. We're very conscious about data privacy. And of course, you have to be conscious about the crosstalk too, because the more crosstalk there are, the more you're opening up your system to threats, all those things. And like Odu mentioned, this is still something that I still hold very dear to me. A couple of years ago, my team and I were awarded the 2023 Public Health Agency of Canada Award in Applied Science and Research. So having said that, I got offered a role a few months ago with the University of Manitoba. They identified some of the work I was doing, and they felt that the Department of Biochemistry and Medical Genetics will benefit greatly from it. So it's a fairly new role. I'm still trying to navigate the intricacies of things. And right now, I'm spending most of my time writing grants because I need money to do some of the research I want to do. What I really want to do is I want to look at rare diseases using bioinformatics tools, rare diseases that affect transplant patients, and in addition, that affect newborn screening, so newborn babies, because having data on the provincial lab has made me realize that there are a few gaps that we can close, where when babies are still, or fetuses are still in the womb of their mothers, there are some things we can identify that will eventually lessen the pressure that it currently has on the public health system. So if you can identify things like deafness and some other things early on, or see how the illnesses or the genetic composition of the mother can influence the baby with or without certain genes. How can we curb that as soon as possible? So that's essentially what I'm doing right now. I have enough to supervise undergraduate students that want to do their thesis, but I don't have enough for graduate students. So maybe some of you are on this talk and you're like, oh, maybe I should reach out for graduate school. Not quite yet. I don't have money right now for that, but that's something I'm working on, funding permitting. We hope to expand my group and do that. And so in addition to all of that, so I partake in departmental seminars and lectures. So there are a few things I'm going to leave you with. Haven't said a lot right now. What has guided me from going from a pure wet lab background to now a pure computational biology background, I would say are essentially three things. The first one is remember your life goals and why you started the journey you're on in the first place. I knew what I wanted from a pretty young age. I knew I wanted to be a researcher. Didn't necessarily know the kind of researcher I wanted to be. Did a lot of wet lab work during my undergraduate studies. Like I mentioned, I ended up working and volunteering in about 7 labs by the time I was done. My PhD too built on that. It made me know what I didn't want to do, but also made me know that research was still what I wanted to do. And I was fortunate enough to find the space in the bioinformatics niche. Then one thing I like to tell people is that know that you still have a lot to learn, even after you've learned a lot. Like I said, I'd learned a lot after my PhD, but I knew I still wanted to learn something completely new to get to where I wanted to be. And lastly, follow your heart. Even if it leads you to new territories, don't be afraid. There are so many people who will tell you they don't necessarily enjoy what it is they are doing, but they just don't want to go through the burden or the stress of starting all over again. It's not easy to start all over again, but I like to think if that's where your heart leads you to, you would have the perseverance, the grit, and the fortitude to keep going, regardless of wherever it is you find yourself. And if you're not fortunate enough to have mentors in your life, I always tell people if there are mentorship programs you can find online, sign up for them. If there's a community like Bioinform Her that we have right now, join such communities. If you have to invest in a course, invest in a course. I think the course I took during my PhD phase, it wasn't cheap. It was very expensive for a student, but I still, invested in myself. And right now, I would say I'm very thankful that I did. And yeah, that brings me to the end of my talk. Oh, yeah, I left one out. The last one is for those of you who are in graduate school, like master's or PhD. I like to tell people that even a master's sometimes is good enough. I encourage people to do, if you want to venture into bioinformatics, I encourage people to do at least a master's because I feel it gives you the tools you need to venture into self-learning. More often than not, masters, you have to be self-directed. During your undergrad, you've been spoon-fed a lot. You need to have that self-directed path because I feel once you can figure out the kind of research you can do, or even draw out questions or objectives from your own research, it would help you down the line to venture into whatever space that you hope to be. And so I would like to thank you for your attention, and I'll pass the ball back to Odun.
Okay, Dr. Ayo, thanks so much. That was like a lot. You unpacked a lot, and I'm very sure people have a lot of questions. They're already dropping it. But I think it would be nice for us to have Lucy do a presentation, then we can take the Q&A section all together. So thank you so much for your time. So I'll go ahead and introduce Lucy. Toby, can we have Lucy biography? So Lucy is actually a research project coordinator at Manitoba Center for the Health Policy, and she holds an MSc in Pharmacology and Therapeutics, also from University of Manitoba. She has a background in biomedical engineering, and she has a unique perspective to healthcare innovation. Interesting. Through her experience, she has come to understand that developing new therapy without fully exploring the underlying causes of diseases can limit clinical success. She's broadly interested in bioinformatics and also pharmacogenomics. Particularly, she's passionate about using computational approaches to study genetic factors and pathways involved in disease. She believes that applying bioinformatics solutions can help guide the development of more elective treatments and prevent deaths from conditions that might otherwise be avoided through genetic research and targeted therapies. Lucy is committed to contributing to research that bridges the gap between scientific discovery and practical healthcare improvement, with the hope of making a meaningful impact on people's lives. So, Lucy, welcome. We'd like to hear your story. We look forward to that. Thanks so much.
Thank you for that introduction. Just give me a minute to share my slides. Can you actually see my slides and you can hear me perfectly?
Yes, we can.
Okay. Thank you again for that introduction, and thank you, Dr. Bolaji, for the very great presentation. Many times it's difficult to talk after someone who has such an amazing bio and someone who is very eloquent and someone who is very experienced in the field. So thank you for sharing that with us. For today's talk, I'll definitely share a little bit about my journey in bioinformatics. Mostly how I got into bioinformatics, a little bit about my graduate experience. That would include a little bit of my research. I won't go into detail for that. And then maybe a couple of courses that I actually took. And hopefully by the end of my presentation, I'm able to actually encourage someone to join the bioinformatics community, because it's pretty interesting and it's a very evolving field. So before I get into that, like she said, my name is Namuli Kevin Lucy, and I come from a very teeny tiny country in Africa called Uganda. Probably a lot of you maybe might know it or might not know it, but this is where I actually did my undergraduate degree that I'll speak shortly about at Makerere University. And currently, like they said in my intro, I work with Manitoba Center for Health Policy here in Winnipeg. And at this center, we host one of the biggest or largest data repositories in Manitoba. And most of the data in this repository is administrative data. And we use this data to actually conduct population-based research in health and maybe some social determinants of health. So my undergraduate degree was in biomedical engineering, and this is essentially a very, very broad field, because, you know, you're trying to pick bits of medicine, you're trying to also take bits of engineering, a bit of chemistry, and, you know, you're trying to encompass all of this in just one field, which is biomedical engineering. So it can be a little bit daunting, but however, there's a bunch of different specializations or different fields that you can specialize in, including medical imaging, bioinstrumentation, tissue engineering, etc., etc., etc. But for me, what really interested me in my undergrad degree was drug resistance, which is actually a topic that you find in bioinformatics. But at that time, I did not really appreciate it as much because, you know, as an undergrad, you know, you're just trying to work through all these so many courses that you have to do. You have to keep up with all these grades and all that. So at that time, I did not appreciate this. So it only truly hit home when I got curious about, you know, how drugs work, right? I mean, day in, day out, we get sick, but we take these things, you know, you just walk to a pharmacy and they're like, okay, take two tablets of this, take 2 tablets of this. And you get fine. But however, that same medicine but can be given to someone else, but this person might not have the same reaction as you might have had. Maybe it might not actually work for them. Or maybe you might find that another person has to take a certain kind of dosage for that same drug for it to work. And of course, there's a lot of factors that actually come into play that, you know, explain all these differences. It could be, you know, how your body reacts to the drug, the environment that you're living in. But one of them that really interested me, that is pretty interesting and is still was interesting at the time and is still interesting right now, was genetics. So if we kind of like backtrack, like for instance, back in the day, without all this modern technology and advances in research or in science, how did people used to actually detect these different genetic disorders? For instance, if you just look at this image here, you have a bunch of people, but some of them are orange. So that means they are different. So back in the day, what they would do, for instance, if you, maybe you would go to like a practitioner then and you have these kinds of symptoms that, they've never actually seen. The disease has not even been talked about at that time. They think to themselves, okay, what is going on? Because when they look at the community, maybe other people don't have this condition. But when they look at your family, maybe your sibling or maybe your parent has the same characteristics, but they still don't understand what's going on. And while they do all this medical history, they find out that maybe you also have ancestors in your family that actually died of the same condition and they did have the same characteristics. So there you can easily conclude that maybe there's something really unique about this family, that is different from other families, that is making them, have whatever characteristics or symptoms that they are having. So this is where, you start to think about genetics, because at that time we did not have all the sequencing like Dr. Bolaji was talking about, the advancements that we have currently. So over time, we see that a lot of data has been collected, a lot of data has actually been curated, and that alone was really, really fascinating to me. And for you to be able to work with this data, because this data has helped us, predict diseases, right? We can now prevent these disorders that, haven't, genetic disorders, we can be able to predict this earlier on. And for you to be able to do all this, you really need a substantial knowledge about bioinformatics and the different tools on how this kind of data can actually be manipulated. So that's what really interested me in bioinformatics. So just briefly, after my undergraduate degree, I stayed as a research assistant and as teaching assistant at the same university. And I had a very good mentor with whom I did share a lot about, my interest in bioinformatics. But at the time, I think this was around 2018 or 2019-ish, if if I have it correct. But at the time, we didn't. Many of these advances are there in these countries, like in the Western world, but in my country specifically, we did not have a lot of these advances there. So all I could do was, attend workshops, where they taught us a lot about Python, about R. But however, I did not have that environment where I could actually be able to practice all these skills. And of course, we know that over time, if you can't sharpen your skills with technical experience or with hands-on experience, with hands-on practice, then all this knowledge can fade. So with all these trainings, I talked to my mentor at the time, and I'm like, well, I'm in this research environment. Yes, I'm doing research. I've been exposed to it. But however, I do not have that hands-on experience. And that's when he suggested that, it would be good for you to do maybe a graduate program or a master's program. So that's how I got to look at different websites. Like, when you're looking for different courses, I went to different websites, looked at their programs, looked out for possible fundings. And one key thing that I can mention here is that if, for instance, you want to do a master's program or a PhD program in bioinformatics, normally that might not be the exact name of the program. So when you go to these websites and you're looking at, the core, you're looking out for the program that you want to take, try to also look at the courses that are taken within the program. Now, for instance, I did a master's in pharmacology and therapeutics, but my research was strictly, or it was in genetics and in bioinformatics. So you might not strictly find that the program is called the way that you want it to be. So it's always good to look out for the courses that are being taken within that course. So that is how I got to the University of Manitoba. And some of the courses that I did were computational biology. I did a lot of general pharmacology because, of course, I was interested in drugs. I did molecular pharmacology. I continued to take workshops and trainings in programming because, of course, if you want to be better at bioinformatics, you need all these skills. You need you might be working on research that requires the use of R or Python or Linux, name it but a few. So for my research, I had to choose between dry lab work and wet lab work. Dr. Bolaji has talked a lot about wet lab work. I think for me, when I got to the program, I already knew I needed the work-life balance. And wet lab sometimes does not give you that balance, like Dr. Bolaji mentioned, because you might be running an experiment and it needs you to, watch it. It has to go all through the night. And, you're running to the lab at 2 or 3 in the morning just, to look at your experiment. I already knew that wasn't for me because I also hated pipetting. Hate is a very strong word, sorry. But yeah, I did not like doing a lot of that stuff. So I talked to my supervisor about this, and then my supervisor was like, well, you're best suited for dry lab work. I mean, bioinformatics has both of it. So for my master's research, I worked a lot with genome-wide association studies, which I'll talk in detail later on, just to show you what came out of my master's work. I also worked with phenome-wide association studies. So essentially for genome-wide association studies, you're literally scanning for markers across someone's genome. These markers are usually variations, and you can associate them with a certain phenotype, or the phenotype might be a disease, or it might just be a general characteristic. And then for the phenome-wide association studies, here you just start with this variation and you try to associate it with different phenotypes of interest. For instance, you might have a certain genetic variation that might be associated with, how tall someone is, or what kind of skin tone someone has, or maybe it's associated with a certain pathogenic disorder. So I worked with a lot of these things in my research, and with my research, I had to build different pipelines, still through either R or Linux or Plink, given the different part of the research. Another key important thing that the master's programs the graduate programs at University of Manitoba allow or give is the bit of networking. And this is actually important. You can't have all these skills when other people don't know them, or you're not interacting with the broader community, because you won't still know what advances are there in the field. So networking is a very important aspect in bioinformatics. And at University of Manitoba, we are given the opportunity to attend both national and international conferences. And here we are able to share our research while we also learn from other people. So like I said, I worked a little bit with genome-wide association studies. So essentially, usually this is how the results are actually presented. So all these dots, this is like sequencing someone's genome from like chromosome 1 to the last chromosome. And each of the dots that you can see there is a genetic variation, right? So what genome-wide association studies do, like I said, you scan for markers or variations within the genome, and you associate them with a phenotype. For instance, if someone has, let's say, a genetic disorder as like Huntington's disease, and then you scan their genome, you want to see what genetic markers in there are associated with Huntington's disease. And you can use this to either predict, when someone can have the onset of the disease, or if someone else within that same family might have that condition. So any dot that you kind of see above this red line is positive or is associated with a disorder. So, but however, when you look at it, you kind of still do not know what exactly or which variation is actually associated. Some of them might be false positives. Excuse me, sorry. Some of them might be false positives. So what I further did for my research in this regard, we used a bunch of fine mapping tools, which are bioinformatics tools like SUSY, still running programs like R and Plink to be able to find which of these dots is actually a true positive. So I haven't really presented much of this data because I did not get the approval to share that kind of information here. So sorry about that. The other one that I talked about were phenome-wide association studies. Oh my goodness, English. Yeah, and I said here what you start with is a genetic variant and you're trying to associate it with, you have a single genetic variant and you're associating it with, like, say, multiple phenotypes. And there's different ways you can, present this work. And still we used a lot of bioinformatics tools, like the locus to gene fine mapping, variant annotation, and still these tools are actually either run in Plink, R, or Linux, or Python. And there's also other programming languages, But what I'm just presenting here is what I actually did for my research work. And one of the pretty exciting things that I had from my master's, many times it's very difficult to get a paper published, but I was able to have a paper earlier this year that was published, and one has already been sent out to a different journal for publication. So that is one pretty interesting thing that I got out from my master's. So I hope I haven't bored you a lot with, this bioinformatics talk or scared you away. But one thing I can say, it's a pretty interesting field, and I would encourage anyone to get in there. So I just always add this slide here to show what a graduate program actually looks like, because many times, you join the university and you're like, yeah, I'm gonna do bioinformatics. I'm pretty much excited. This is what I'm going to do. You think it's like a very straightforward field. I get in, study, do my research, and live. But many times this is what happens. You have a bunch of like unrelated projects, and you just see your project fading. But then you also have to produce. But the takeaway from this slide is that with a graduate program, you're not just taking the technical skills from there. You're picking up also other transferable skills or even other technical skills. So this unrelated project is one that can maybe give you the experience with other, should I say, pipelines, or with other genetic data sets that you might not be working with in your primary, research. So it might be daunting sometimes, but it's we all go through it, and there's always like light at the end of the tunnel. So okay, sorry, my screen is, oops, it's frozen. Okay, yeah. So I'll just get to my acknowledgements. This is my lab. My supervisor is in there, and these are the people that I worked with. And of course, I wouldn't have been able to get this far without a lot of these funding bodies, because, when you get into the program, you're able to you're able to apply for extra funding. So I know funding is a very important and key thing here, because education is pretty expensive. But when you get into the program, in almost all universities, I think, then other opportunities can open up for you to apply for extra funding to actually support your studies. So after school, then what? How do you build your your career? How do you use all these skills that you've picked from your graduate program, both the transferable skills and the transactional skills? How do you put them together for you to build your career, to, you know, apply for a job and, you know, you and get the job? So one of the things that I can always that I can say is that continuous learning is very important, because like I said, this is an evolving field. New technology is coming in almost every year. There's like a new bioinformatics equipment, right? So if you just lean back and you're like, well, I'm just going to stick with my R skills, I don't think that will be very developmental. So there's a lot of things that are coming up. I mean, there's a lot of also machine learning, there's a lot of AI, different sequencing tools. So try as much as possible to continue learning. And you can do this by attending different workshops, attending webinars, attending data camps, because in these spaces, this is when you're able to actually interact with experts in the field, and they are able to inform you on, you know, the emerging trends and telling you or letting you know of, you know, the new things or skills that are on board, or the equipment and the like. And in these programs, you can, of course, better your skill set in programming, because it's a very important skill in bioinformatics, data visualizations, and still using those different bioinformatics tools. And when you get all this, you have to build your portfolio. You can create your portfolio on GitHub repository. I think Bioinforma has had a webinar on GitHub. I think so. I could be wrong, but I think I've seen one of those. You can always also post your work on LinkedIn, of course, if it's acceptable, because many times this is research, and it might have a couple of copyrights based on the university. So you can't just post that stuff on different pages. But if you get the green light, try to post your work, because it makes you visible within the community, and try to also contribute to the open source tools. The last one you can think about doing is networking. Attending conferences, like I said, is a very important thing, and don't just stop at attending. Always either have a poster presentation or an oral presentation, because this is when people can actually approach you about your research. Because if, for instance, someone in the audience or someone who has interacted with you at your poster is interested in your research, or they have a similar research that they are doing, then definitely they are going to discuss it with you. And, you know, that's when, you know, you build your community. Join professional organizations, of course, and online communities. Oh yeah, that's pretty important, because there's a very, very big online bioinformatics community and a forum. And these communities are very, very helpful. I think the best example I can use in this case is in my program, I think there was a pipeline I was running, and I had this era that it was going on for 3 months. I couldn't do anything. But the minute I posted this on an online community, someone had a solution for me. And I'm like, oh, why didn't I think about this earlier on? So my point is, when you run into an error, there are high chances that someone out there has also had a similar error. So if you join these online communities and make this public, then someone is able to actually help you in a timely manner. So after building your portfolio, there's different career paths. I believe Dr. Bolaji has talked about almost, like, should I say 80%? I feel like she has a very good bio, really. She's worked in very many places. So you can either work in academia or industry. In academia, you can work as an educator. You know, you can mentor the next generation. You can conduct research. There's different types of research or different types of data that you can work with in bioinformatics, genomics, transcriptomics, proteomics. So all that you can have you can work in those different fields in academia, and then you can also work in industry, big pharma companies, clinical, you can be a clinical bioinformatician, or you can even work in a software company because you already have these coding skills. So this is not all. There's a lot of places that you can work in, but I just shared a couple of examples that you can go out to. So if you haven't got a lot from my presentation, these are maybe the takeaway messages that I have. Always get started somewhere. Everything might it might seem daunting at the start, but it's very, very possible. Do not try to learn all the data types or the tools at the start because you might be discouraged. Because like I said, there's a lot of different types of data. You can work with proteomics, genomics, transcriptomics, but all these are different types of data. Now, for instance, for me, I used a lot of genomics data, but the skills that I learned with working around genomics data, I can still transfer them to the other the other data sets. But if you try to learn all this at a go, it might really be daunting and it might discourage you. So try to learn one at a time and try to use the skills that you learned from that one to work with another data set. And then, like I said, try to show your learning journey and get a very good mentor. Thank you. I'll just stop
Thanks
sharing.
a lot, Lucy. That was so nice. That was so interesting and well detailed too. Thank you very much. We are over time, 10 minutes. So we will only take, I could see people already raising up their hands. So we will only take 2 questions and we have some questions in the chat. And I think to be nice, we give them the chance first before. Yeah, so I will read 2 questions from the chat and the other ones will be sent to our guests. We'll get response from them, then we are going to share their response with you via email. So, I mean, any one of you can actually pick up the question, but I think this particular question was dropped after Dr. Ayo Oluwa's presentation. It says, it's from David. It says, thank you for your presentation. Do you necessarily need a domain knowledge in biology to transit into bioinformatics? Can one start from the computational part and work backward to a certain domain?
That's a pretty good question. It's a very good question. Before I answer, I also just want to give a shout out to Lucy. Very good presentation. And I think I'm even going to draw from one of the things she said to answer this question, where she said start from somewhere. So if that's what this person wants to venture into, I would say start from somewhere, because I'm assuming this person maybe already has some computational skills or some computational parts to it. If you want to fully venture into the bioinformatics space, start from somewhere in terms of there are publicly available data out there. Take some time to understand those kinds of data. So if you want to do maybe cancer, if you want to work with cancer genomics, read 1 or 2 cancer papers, understand some of these genes, what it means for them to be upregulated, downregulated. And that's because those are some of the things a non-biology or a non-health, life sciences or health sciences background wouldn't have. But you can at least get the full gist of it so that you understand what it is you're doing with your data. That's to say that it doesn't mean if you don't have the time, you can't still have a space in the bioinformatics space. So as it's been said, I work in the National Microbiology Lab, and I've had the chance to work with pure computer scientists, those who have their background in software development or software engineering. The person I worked with that helped us to develop our NGS laboratory information NGS database. She has a background in computer engineering. She's a data architect, and we're able to bring her on board, forwarded a couple of papers for her to read just to understand what it is we wanted to do. And then she was able to help us. So what I'm just trying to say is that you can work from the computational side and work backwards, like you've said, you can work backwards by reading papers, understanding what it is your data has that you can then use your strength to draw from. Oh, I think you're muted on.
I'm sorry. Thanks so much, Dr. Ayo. Lucy, would you like to add something?
I think Dr. Bolaji has said it all. You always have to start from somewhere, right? You can't, it's just the way I think about it is someone told me this joke of how would you eat an elephant? It's very big, right? But you
Yeah.
gotta start with a small piece at a time, picking it up at a time, one at a time, one at a time, until you get, something done. So it doesn't really matter whether you start with the computational side or whether you start with the other side, really. I mean, I have an engineering background, right? I had a biology side, but I did not have the IT side. So that's where I started from and went into, the computational side. So, but then we have a lot of people that started with, studying computer science, computer engineering, but they don't have the biology side, right? So, but over time, they also invest in the biology side because they want to be very good at bioinformatics and what to do. So like Dr. Bolaji said, whichever side you start with, just make sure you invest the time, you invest your time in it. Read a lot, join those spaces where people share these kinds of information, and yeah, I believe that would be very helpful. Yeah.
Thanks so much for your response. I'm I'm thinking which one to pick, but I think let's go for this one. I think this is a personal question. Someone dropped this saying, "What step will you advise someone in with a master's in bioinformatics, 7 years post graduation, but no related work experience since graduation, but would like to get back in the space? Is a PhD needed?" Would you like to go?
I can, oh, Dr. Bolaji, you can go fast. That's fine.
Okay, thank you. I was going to say that a PhD isn't needed. So the person is asking, is a PhD needed? Personally, I don't think a PhD is needed. This person also said they have a master's in bioinformatics. So this is telling me that they already have the technical skills. Even if they've not used it, it's there. It's like riding a bike. Once you know how to ride a bike, even if you've not gone on a bike for 20 years, once you see it, yeah, you might be rough in the beginning, but you will get the hang of it. So I would say for this person, just again, like what Lucy has said, try to get your hands on some data and put your stuff out there. So get a GitHub page, post it on LinkedIn, tell a story. So if it's, you want to pattern a data from inception to completion, develop your figures, even if it's to submit it to bioarchive, let people see that you can start something, you can identify the objectives, and you can draw figures, make figures, tell a story, write something small, put it out there. And I feel from that, because even when I've had the opportunity to hire people, when they put their LinkedIn or GitHub page, we go and check. So people would go check, see what it is you've done, and that would be a nice way to get back into the space. I would let Lucy add any other thing she wants to add.
Just the one thing that I can add, of course, you might, yeah, like Dr. Bolaji has said, the PhD might not be necessary. Do the PhD if you want to, because a PhD is a monster of its own. So do it if you want to. But otherwise, there's different or other avenues or resources that you can actually use to sharpen your skills, right? If you already have the bioinformatics skills, but however, you haven't been in the field for 7 years, one, go out there and find what new technology has been published, what new data types have been published. And when you find this information, you can take online courses. There are so many online courses out there that don't need paying for, even like some of these resources are out there. So you can do that to like sharpen your skills. And if you're looking at applying for a job still in that field, the way you can tailor your application, you can be like, yes, I haven't been in the field for 7 years. Yeah, I understand that. But however, I have taken these courses. This is what I've done to sharpen my skills in the field. And I'm very much suited for the position. And yeah, of course, you have to show this somewhere with a, like, for instance, a GitHub page, like Dr. Bolaji mentioned.
Thanks so much for that. I mean, I would like to like ask one final question. This would be like your final gift to them. So I think everybody needs this because one of the biggest limitation in life is what we think, our mindset. So this is from Jamil. He said, "Hi, Dr. Bolaji, but I think I'm addressing this to both of you. Great presentation. In your opinion, what are the skills and mindset changes that are most important in developing a successful career in computational biology?"
Uh, yeah, that's a heavy that's a heavy question. I'll start with the skills, and I think for the skills, it depends on the stage the person is at. So if you're talking to someone who had just finished their undergrad, the skills they're going to need to get into the space is different from someone who has a master's versus someone who has a PhD, and then also depending on what exactly did you do these studies in. But I would say generally have an open mind. Like have an open mind. And I think one of the beautiful things I like about what Lucy also said was that there's always something to learn. You can't say, oh, I already know this, or I'm an expert in this particular kind of data, and forget about the others or shy away from the others. So having an open mind has helped me. If you followed my talk closely, you would see that I transitioned from, plant interaction to microbes, then to, animal viruses, and now I'm working with human pathogens. So once you have an open mind, you would know that whatever task is thrown at you, or whatever challenge you see, you want to work on it. You're not shying away from it. Then also the mindset of always learn. I say this over and over again, and it's something I try to live by. I try to learn something new every day. Once you have that mindset of learning, 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 there's so much you can learn just by reading, watching webinars, watching even stuff on YouTube. As powerful as all these social media tools are, if you can find what it is you're looking for, it's not always a time waster. If you find, a pretty good podcast that you follow that is all about bioinformatics or all about, computational biology or data analysis, there are one or two things you can pick up on that you also want to, transfer to your toolbox. So just so I don't keep on talking, there are so many mindset changes, but I will leave you with those two, learning something new and also knowing that there's so much you can learn out there and having an open mind.
For me, I'll just add on what Dr. Bolaji has said. Learning. That's the one thing that comes to my mind, because bioinformatics it's an extremely, it's evolving at a very, very, very fast rate. So if you don't have that mindset or the willingness to learn new things, then you won't survive in the field. So you need to be able to have that mindset change of, I'm willing to learn new things. It might be daunting at first, but I can still do it. So yeah.
Thank you so much. It's been an amazing section, and I'm very sure everybody here has learned a lot. I can say one or two things a lot. Me especially, I've learned so much. So thanks so much for honoring our invitation and for taking your time to actually come up with such great presentation. You didn't only tell your story, you actually help us to draw out the lesson. I think that's the most important part, and we really appreciate that. Thanks so much. I want to ask, can our participant follow you online on LinkedIn? Are you open to connection? So please feel free to follow Dr. Bolaji and Lucy online. And if you have any question, further question, you can send it to us, or you can, I'm sure, if you chat them, they will respond to you. Thanks so much, everyone, for coming, and we truly appreciate your time. We took a lot of your time. We're sorry, but thanks so much. We really appreciate. So this is how we are going to wrap up the section for this quarter, for this webinar. And I'm going to hand over back to Toby. Toby, are you in? Are you still around? Hi, Toby.
Hi, hello. I'll just say a very quick and deep thank you to Dr. Bolaji and to Lucy. These sessions were very, very inspiring to me personally, and I'm sure to everyone in attendance. As I can see from the chat box, a lot of very, very positive responses and messages full of gratitude. So that gratitude is also what we want to express. We are really grateful for coming around, grateful for sharing your experience, and we believe it's actually achieved the goal to have bioinformatics enthusiasts inspired and to get some more guidance as we build on our careers. And with that, we say thank you and we look forward to another webinar sometime soon. Yep.
Thanks, everyone, and feel free to leave. We have another section with our scholars after this. So thank you.
Thank you for the invitation as well.
You're welcome. Thanks for honoring it.
Yeah, thank you very much for having me.
You're welcome, ma'am.
Thank you.
Bye. Have a lovely day. Yeah. Mm. Okay. I'm still waiting for people to leave so that we can start our section. Okay. I think I can identify some scholar. Please, guys, could you actually rename yourself? Just go to your what's the name? Go to your name under participant and click on the three dots in front of it, and you're going to see rename. Please rename yourself properly so that I will know who and who on this call and who is missing. Thank you. I think we can do that in the next 2 minutes. Yeah, 2 minutes is very much. Go to the three dots, put the cursor on your name, go to the three dots in front of it. Please rename yourself. Garima, no, Olama, please, can you do that? Okay, thank you, Olama. Hmm. Oh, really? Okay, let me just check this out. Okay, so welcome everybody. Thanks for joining this section, and let me say a warm congratulations to you today. It's Can I say it's a privilege? It is actually to be here and to be among the midst of amazing scholars. When we went through your work, I always heard that a lot of you did a very good job. I mean, you didn't just do the project and dump it on us. I was really amazed at the quality of the reports. It was so detailed. A lot of people, I would say more than 90%, gave a well-detailed report, and that is really amazing to us because one of the things is you need the skill, but you also need to be able to present your result well because that's the only way you can sell whatever you have that will convince people to know that you actually know your job. So well done, guys. You really did great. And that is why you're here today. Unfortunately, I think two tough people are not here, but well, maybe we have about 30 minutes. Maybe we could briefly introduce ourselves, tell us your name, where you're from, and what you do. Are you a student? Anything that you do, briefly. So, will you go ahead, Akbara Chita, please, Jita, please. I'm so sorry if I'm pronouncing your name wrongly. Please, feel free to correct me. Will you go ahead? Chakraborty, are you here? Okay, I guess she's not here. Babatunde Akibo, will you like to go ahead and introduce yourself? Can you guys hear me? I'm not getting any response from you. Okay. Okay, let's go with people that have responded. Moyosore, do you want to introduce yourself? Moyosore.
All right. Hello.
Hi, good evening.
Good evening.
Yeah, please go ahead.
Sorry, ma'am. I could not hear you at first. There was some problem. Could you please repeat?
Oh, I was asking you to introduce yourself. Can you introduce yourself? What your name, what you do, and where you're from?
Okay. I am Aparajita Chakraborty and currently I am a PhD scholar. I'm pursuing in the field of biotechnology. I'm from India, St. Xavier's College in India.
Okay, nice to meet you.
And I'm actually very much interested in bioinformatics, so that's why I actually took part in this particular project which had been hosted by Bioinform HER. So it was actually, I really enjoyed doing the project, and actually I'm really interested in the field of bioinformatics, so it was actually a very nice experience for me. So I would like to thank all of you for giving me this opportunity to participate in this project.
You're
Yeah.
welcome. Okay, we move on to Bashira.
Yeah.
Dr. Bashira, do you want to introduce yourself? Okay, Garima, will you like to introduce yourself? Dr. Bashira has a problem with her mic. Okay. Garima, are you there? Will you like to introduce yourself? We want to make it very fast so that we can end this meeting latest by 7 p.m. of my time. That's 6 p.m. West African time.
Yes, my name is Garima. Currently, I am pursuing my master's in biotechnology, and I am very excited to join this webinar on bioinformatics as I love this project also. I enjoyed very much.
So, Karima, where are you from?
I'm from India.
Oh, okay, nice. It's nice meeting you. Glory, would you like to introduce yourself?
Good evening, everyone. My name is
Good
Glory
evening.
Jayoba. My name is Glory Jayoba, and I'm from Nigeria. Currently, I work with the Nigeria National Center for Disease Control in Nigeria as a scientific officer. So I'm really grateful for this opportunity to learn bioinformatics because I'm really interested in advancing my skills in bioinformatics so that I can also be applied also in my field and my job. Thank you.
You're welcome. Thanks for introducing yourself. No, am I correct? NOE? Would you like to introduce yourself? Okay, no response from there. Olama, would you like to introduce yourself?
Okay, good evening. Can you hear me well?
Yes, good evening.
Okay, so my name is Olamma. I'm a Nigerian living in Nigeria. I recently graduated as a microbiology student. You're smiling.
Yeah. Yeah.
Okay, so I recently graduated as a microbiology student in 2024, and currently I work as a quality intern in the food industry. So my work revolves around quality assurance, quality maintenance, and quality control. And I'm hoping to transition into computational biology, which is why why I was actually interested in participating in the bioinformatics project. I still very much love wet lab research, but the way the world is going, I can see that there's a need for more scientists who have computational skills, and that's why I'm here. Thank you for the opportunity.
Thanks so much for introducing yourself. Shaiti, am I correct? Would you like to introduce yourself?
Hi everyone, I'm Sahiti. I'm from India. Currently, I've completed my master's in biotechnology and I'm looking for more opportunities in bioinformatics as I want to shift into that field. And in future, if possible, I would like to pursue, I would like to get a PhD in the very same field. And thank you.
Thanks so much, guys. Thanks for introducing yourself. I'm sorry I didn't introduce myself. Oh, Moyo,
Yeah, please, you left me out.
please, please go ahead.
All right, thank you so much. My name is Moyo Sore. I am a master's student at the University of Medical Sciences here in Nigeria, studying plant genetics. I'm very much interested in bioinformatics, and which is why I am here. And I'm grateful for the opportunity, and I look forward to learning with other students as well. Thank you so much.
Thanks so much. Sorry, guys, I didn't introduce myself. I did at the beginning of the previous section, but right now, let me give you a brief introduction. So, hi, my name is Dr. Debi Sefia Todonla. A PhD student at Stellenbosch University in South Africa, and I work on spatial transcriptomy. It's actually a branch of bioinformatics. It's more like your RNA-seq. I know a lot of people will be familiar with RNA-seq, but it's not really RNA-seq. It's more like you're looking into the space as well as you're looking at single cell. So it's a bit, yeah, complex, not that much. So we have Toby here, who is the founder of Bioinforma. Toby, would you like to introduce yourself?
Hello, everyone. Yeah, so my name is Toby, and I am very grateful to hosting and having you guys around. I am currently doing a master's in physiology and pathophysiology at the University of Manitoba, and I also, just as you are, an enthusiast of bioinformatics, and I look forward to having you guys around. Thank you.
Thanks so much, everyone. So just to make things briefly, I'm going to tell you what we're going to be. We are not doing much. I'm just going to be sharing some information with you. So for the art training course, it's going to be for 2 weeks. Sorry, 2 months. 2 months. It's going to be for 2 months. That's 8 weeks. And we're going to be starting in July. It will be starting July 1st. And I'm going to put everybody on WhatsApp. Yeah, I think I have everyone's contact. I'm going to add you to a group. So if you find yourself in a strange group, do not be scared. Yeah, but of course, you will clearly see our name by informal. So we are going to have 22 scholars in on that WhatsApp page. So you should look forward to that. And for this training, we are going to be using a platform. We are going to be collaborating a bit with Akbar. So we are going to be making use of Akbar our course, and we are going to give you access to the platform once the course, before the course actually begin, before July 1st. I will make sure I send everybody access to the platform. So on the platform, there are modules on it, and for each week, we are going to tell you which module you have to complete for that week. And every weekend on Saturday evening between 7 p.m. West African time to 8 p.m. West African time, we're going to be meeting to discuss what you've learned from that particular module. And everyone will be showing us what's the name? A platform, because we want to make sure that you're actually following the group, you're completing the module as you're supposed to. And we having that WhatsApp group is an avenue for you to ask questions when you get, maybe you get stuck at any point. So you're allowed to ask questions in that group. And of course, you are free to like chat either me or Toby. If you are doing your analysis and you get stuck, we will be willing to help. And during these weekend section, you have the opportunity to ask questions about the module for that week, or even anything outside the module that you're working on, maybe in your own personal space. Yeah, so basically that is how we're going to run the program, but you should have it at the back of your mind that by the beginning of August, which is 5th week, you're going to be having projects. So we are going to be having like about 3 to 4 projects, and I expect everybody to complete their project on time. Because if you do not complete your project on time, what's the point of being here? And if you do not complete your project on time, you cannot graduate from this program. And also, you won't get a certificate from us. So we expect you to complete your project. And you all know how we do our projects. Whenever you do a project, you are expected to put it on GitHub because we want you to have a good portfolio in such a way that recruiter can actually check you out at any time and say that you're good in informatics and beginning to employ you. So be ready to write more detailed reports in the next 4 weeks or 5 weeks to come. I know that you guys did well, so I'm not worried about report writing. So I don't think we are going to be treating report writing at all because you guys are good in that aspect. But I want everybody to be serious with the cause. This platform, you're going to have access to it for a very long time. So even if you finish the course, you have the opportunity to go back and look at the course material. You look at the course material. And one thing about the platform is it's a combination of text, video, and also papers outside the platform. It will direct you to paper outside the platform. So for that, I expect that you don't just watch the videos. Every weekend when you call me for our section, you are to come with your script that you use to practice whatever you learn from that video. So I want to see what you're doing. So keep that in mind that every weekend you need to show us something. Your progress, that is the platform. The second thing will be the script. You have to show us your R script that you actually ran for that week. So, I mean, it's not hard. I know that maybe some of you are totally new to R. R is not that hard. I've been working with it. You just need to understand the basic syntax, more like you understand the basics of the language you're speaking. If you speak English, you know what is verb, what is noun, what is pronoun, how to join them together. And if you guys are very committed to this, Garima, we've not created a link. I'm going to add everybody to the WhatsApp group, so you don't need to worry about the link. And if I cannot add you, I'm going to send you an invite. So do not worry about the link right now. So all I'm asking from everyone is to be committed to this program. It won't take your time. I mean, you can always, even if you're busy during the day, maybe you're doing school stuff, in the evening, you can always take your course material and read. And one thing about R is once you start it and you're getting it well, you're going to get stuck. You won't want to leave because it gets interesting along the way. So I want you guys to be committed for the next 8 weeks. Yeah, just 8 weeks. And before you know it, you become R Pro. So basically that is it. I don't know if Toby wants to add anything. Toby, is there anything I'm missing that you'd like to add?
No, I think you covered it pretty well.
Okay.
Yeah, we'll be in
So,
touch.
yeah, so you should be expecting your access to Akbao, our database. Sorry, somebody sent a message. Just let me have a look. If you want to say something, you are free to indicate and unmute your mic. Will you like, yeah, please go ahead.
Actually, in the mail which I had received, so there was something mentioned about the scholarship. So if you could elaborate or explain about it in details, that what the scholarship is
Okay.
about.
Okay, so the R material that we are providing for you is not free. It's a paid course material. So Akbao has this well-detailed R course, but it's for sale. It's for $20. So we are giving you scholarship to actually access that particular R course. And you're accessing that particular R course forever. So meaning once we buy it for you, it's yours. So you can always go back to it. So that is what the scholarship is referring to. It's actually not like stipend that you spend in school. Do you get that?
Yeah, okay, thank you. I got it.
Yeah, okay. Is there any other question? I guess we are clear then. If you have a question or you have a comment or anything, you're free to indicate, and yes, the same thing. Okay, in the absence of that, I think that is it. Thanks for coming to this meeting, and be on the lookout for our next email by the end of the week. Your certificate, as well as the link to the course material. Yeah. Okay, thanks everyone for coming. I really appreciate your time.
Thank you, ma'am.
Thank you very
You're
much.
welcome. You're welcome.
you.
Thanks so much. Yeah. Have a lovely evening or morning or afternoon. Yeah.
All right. Thank you so much. Bye.
Bye.